In today's session, we will be recapitulating all the things we did so far, and we'll be talking about data access. We'll be talking about the project abstract, and also some relevant matters related to how to create tables. We will go through those things again. What are the methods we used so far, and how can we combine those methods and access the right data that would be appropriate for using those methods on it. So during the whole session, you can ask us anytime if you don't understand anything or you forgot anything that we did. We can also discuss your data and your project proposals. As far as I saw, there aren't any proposals being typed in the form yet. So I think Mohit can tell us if someone has subjected the proposal to this moment, but I think none of you actually did. But some of you are actually working on your own data sets. So I understand if some of you have confidential data sets, you can keep it that way. You can communicate with us in an extent that you want. But if some of you have some publicly available data sets that you would also like to discuss, you can do it freely during this session. So let's discuss what we did so far, and let's discuss some matters related to the data sets. What would be relevant data sets for you, and maybe we can then draw some conclusions and make some project abstracts and plans on how to continue. So at any time you can unmute yourself or you can post a question in the chat. So I'm sure that this wasn't long time ago when we actually did summary statistics, but we covered a lot of things after that. So maybe it's a good thing to maybe focus on this a little bit and some properties of the tables and the data that we need to ensure to be able to continue the analysis. So I'm sure you remember these pipelines and combination of using P-Info platform, Excel, and RStudio. So we created some pipelines. You can actually see one of them here in the top part of the image, which use that SVL file or FASTQ files. Elia showed you last time how you can use this pipeline on any data set that you can find, for example, on NCBI that is publicly available. So he showed you how to download FESTA file, GTF file, and the NCBI run table. So you upload those three and create a pipeline like this. In the end, you would get an expression table. But the very important step is how to manage that table that you got. What is the type of data that is contained in the table and how do you organize? So, for example, I will just lower this for now and open one of the data sets, for example, in the notepad. So here we have a PAM50. We talked about this data set, right? And we said that this is how TXT table, it's not a table actually.
Yes. This Windows not enough.
Can you please unmute your microphone if you're not asking a question? Okay. So you can ask a question anytime. I see a question from Vamshi here. I have labeled data. Okay. So okay, Vamshi, since you sent this to me, we'll speak about it later. Okay, but for now we'll speak about general things related to tables. But I will answer that question later to you. So what is the thing here? Here is the txt or tab delimited format. If we open this in Excel, we could have different aspects. For example, here you can see that my data is presented as some dates, right? Because Excel is programmed to read many data types, many data formats, not just that are related to biometrics, very used in many other types of analysis, not just so here on how to, I don't know, percentage dates as well, currencies, number also has the option. What we want is our data in as text number. We want to separate this or data type. For data types, we said that those numbers for discrete data, I don't know, from scale to scale from different scales, and these scales are important when analyzing. Now I will show you, for example, how Excel to import table. For example, just a sheet here, right? Two more, and you have the option here, data. You click data, you go to importing data from txt file here. We will get a similar box to the one we're getting in our studio. The important thing here is that you have several options. You have option to change encoding. So for now, we won't change this. We'll leave it here and go to next. You have the preview of the data here, right? So you can see how your data looks like. It looks much better than the first time I opened it. So you can see that it's not in dates. It's actually in uh, in numbers in a format that we want it to be. You also see that our separator or our decimal separator is point. That's very important because decimal separator could be uh, just comma, it could be point, and you also have different options here on how to qualify text. So you see, for example, the text could be qualified as something that is between these interpunction signs. So for now, I'll just keep this to none because it will read the text as text if it's not numbers, so that I don't need to specify them with these. We're reading data from a tab delimited file, so it's important to leave this to tab delimited. And here we have those options on how to treat our data, how to import format of the data either as a date, as a text. You saw that it imported it as a date automatically when we opened that file, but I will keep this to general now. Sorry, I'm just gonna do this again because there is another important step here. So I'll just repeat everything, go to next. This should be to none, text qualifier. and delimiters to tab. So here you set this to general, but we also have some advanced options here. And this is very important because our separator is set to be comma here, right? It's not point. It's not as if we were to import data like this. We want it to be like this. Even if it's like this in the preview, it will not work well if we leave this here to come, right? So in order for it to be the point, we need to type that point here. But now we have another problem. Thousand separator is the same as decimal separator. So it might actually confuse the software. Uh, Software is very efficient, but only if we give it good instructions. So in this case, let's put this to come. I know that we don't have thousands in our data set, so I can set it to comma, and you will see that it won't read them at all. It will just read decimals. And that is generally the situation that we will have. We generally don't need thousand separator in more than, I don't know, 99% of our analysis. We just need decimal separator, and the more decimals we have, software calculates it automatically. We don't need thousands. So this is a setting we should use, and you will see that after clicking OK, it imported the data. But another important thing happened here. It imported data well because our data, 1050, contained points as decimals, right? And I use that option to import this. If I use the option to import commas, it wouldn't import it well. So I needed to use points as import option. But after it's imported, you can see that it's automatically created with commas. So Excel has many options. You can use commas as decimals, you can use points, but this default option is to create it as the commas, as you can see. And in Excel, you can use this table to analyze. I said that we have the data analysis button here, and we can, for example, I don't know, we can run descriptive statistics. We can select this input range and select labels in the first row and run summary statistics. So we can get those values here for these two samples normal likes. We have means of 5.5 and 5.4. They're very similar. We have standard deviations, medians, and other parts of descriptive statistics. So you saw that you can use Excel in a very efficient way. This was something that happens when I, for example, first time clicked on the data set just to open it. But when I use that import function in Excel, everything worked out much better. So we also said that uh, decimals are commas here. We can, for example, save this file. I will save it as PAM50, I don't know, txt. Okay. So let's see how it looks like. Sorry, we need to save this as a txt file. We can save it as an Excel file, which is pretty much the same thing that we would get uh, as the thing that we are looking, as the file that we are looking in this moment. But let me just save this as a txt .txt, okay, and let's see what we created. PAM50 txt. So if I open this with Notepad, you will see that table is actually converted to a tab delimited file. Right? So what is tab delimited file? If I type something here in the Notepad, you can see that, I don't know, it's not tab delimited, right? We have spaces everywhere. But here, for example, from this normal like to this normal like, there is nothing. There is a tab that separates. And between each two columns, we can see that there is a tab that is separate. So that is a tab delimited file. Another important aspect here is that we have commas here. So for example, tbioinfo platform uh, will function best if you provide it with a txt file or tab delimited file that has points as decimal separators, not commas. So very easy way to convert this table to a table with points is actually to do this. You just go to edit and to replace. So you find those commas and you want to replace them with points. So you just type those signs in these spaces here. You have instructions for everything, find what and replace with. So you just click replace all and you will see that actually everything is replaced now and we have the file that could be saved again. So this is the type of file. I just need to close it here. This is, for example, the type of the file that we could upload on tbioinfo. Before we go there, let me just show you another thing that is very important here. Sorry. Uh, wait a second. I will just open this one in Excel. Okay. You'll see that again, same thing happens. Okay, so I'm just going to repeat the whole process here. But we have this file that is saved with the points. But I will show you another thing here. You can do this very quickly. So after these steps, there are some other ways to manipulate the table. To manipulate it, not to change data, that is very important, but to just change the order of rows and columns. So for example, you can consider this situation. Let's say that you want these parts of the table that are actually classes in this case, and these parts changed so that you have genes on the top and classes here. So you just do this. You select everything. Sorry. You just select everything and copy. Now you can create another sheet here and just paste it, but not paste it here or here, which would be a normal paste. You paste it as transpose. So you can see now that it's changed. You have classes here and you have genes up there. Why is this important? Because sometimes you will need to transpose data to analyze different aspects of your data set. Sometimes your data uh, will be in a format that you're analyzing comparing genes, but you don't want to compare genes, you want to compare these. So you have to transpose them back here and compare. So that's very important. You need to have a good format. It's a txt tab limited format, essentially with points to be able to upload it to tbioinfo platform, for example, and run a PCA or some other analysis. When we are there, for example, let's upload one of the files for the PCA. So let's take any of the data. We have those data sets here. For example, we have, I don't know, 1000 cell line, 1000 genes. We have sample names, we have gene names, and the values of gene expression. We can see that it's not normalized, right? So this is another thing that I want to repeat. What is the difference between normalized and non-normalized data? So we have, for example, we have some zeros. We have values that are 1, 2, 5, and then we have, when we lower down, we will find values of, I don't know, 100, 700, several thousand, right? So they're big and different scales in our data. Data is not dense, it's not condensed to some smaller scale. So we don't have a continuum between values. We have big gaps in our data. And that is typically a non-normal data. It doesn't have a normal distribution. When we were analyzing summary statistics, we were mentioning this bell-shaped curve. So a data that has normal distribution typically would be plotted like this on a histogram. So smallest values would be here, and then as we go to some medial points, most of the values are close to that median point. And then we have lowering of the bell-shaped curve, and those highest values, those most extreme values are here. So generally, a normal data distribution looks like this, but if our data is not normalized, it would look completely different. Let me just show you, for example, how normalized data looks like. You can see that there are no big gaps in the data. It's from 0 to 10 compared to the data that we were looking right now. You can see that it's not really the case. So we have, for example, let me just show you some parts of the data. You have, I don't know, take a look at this gene. It's pretty much in expressions from 100 to 500, right? Then we have some zeros here. Then we have several thousands. We have different scales, but if we look at the data set that is normalized, you can see that it's pretty much on the same scale. There are no big gaps in the data. And that is one very important aspect of data analysis. You need to have normalized data for some machine learning methods. For others, you can use a non-normalized data. So please remember that it's not always the rule that we need to normalize it. Sometimes it's actually better not to normalize, but in a large number of cases, we want to normalize. For example, if we run a PCA or k-means, you remember the k-means algorithm that we used in clustering, you remember the PCA, which is a dimensionality reduction technique. Both of them require you to have normal data. What happens if you don't have normal data? The analysis will still run, but your results will not be valid. So some machine learning algorithms require you to have normal data. And in most of them, you can use normal data. But there are also algorithms that can use non-normal data and even in a better way. And we'll speak about that later. So let's just cover some other aspects we did. We run some summary statistics in R. You remember it was very simple code. You just import the data set, which was done here. You use the same options, similar options that we used in Excel, just we had that option to set header as yes. And when we import the data set, it looks like this. So then we use the summary code to create summary or descriptive statistics similarly to the data analysis toolpath in Excel. So here we have means, we have medians, minimums, maximums, and other parts of descriptive statistics. So one thing that I also want to repeat is then that when we opened RStudio, we sent you that video about interface sections of RStudio. So we have this section here, which is a scripting section. You have the environment section, file section, and the console section. So as you can see, in the scripting section, we are writing lines of code that we want to run, and we have the run button here as well. On the other hand, we have the console, and everything that we run here in the scripting section is executed in the console. So we can also view results in the console, and the code is executed there as well. Difference between scripting and scripting section and console is that in the scripting section, we don't actually need to run anything. We can just write code, and there is something very important in RStudio because RStudio is an integrated development interface. So it's not just a coding language. Coding language is called R, and RStudio is an extension that is actually including integrated development tools like code diagnostics. Code diagnostics also use machine learning, which is very interesting, to, for example, propose code or names for us. So if I type just sum here, it will propose the code for summary, right? So there are some facilitation tools in RStudio that actually speed the whole process. Also, if we have errors in the code, we will have a red circle here appearing. We have the file section. Now you can't really see here, but I will close this. I will close everything and open RStudio to show you how to just repeat the whole process of using the file section. So we have, for example, we can go to these three buttons and select directory we want. For example, I can select any directory, for example, DCIS. Sure you remember ductal cell carcinoma in situ dataset that we were analyzing, and for example, open that directory. We can set that as a working directory. For example, in open files, we can run R codes and save files in the same directory and many other things. We also have a terminal here, so I showed you how to run the code terminal. For example, I don't know, I'm just going to open one terminal here to show you how to run some code, but before I go there, I will actually create code from the server key by button. So I don't know, for example, unsupervised analysis. This DCIS train data set, you saw upload simultaneously at the same time. So the important thing, you just reload the page and the problem will be solved. So let's upload the same file and wait for the file to be processed. So we can create, for example, cluster in Python. We said that we have many linkage patterns there that we can use. Let's use averaging, and let's use, for example, Manhattan. Now I'm gonna use the transpose. You have the same transpose option here. Very important. This thing essentially does the thing we did next with copying data and pasting, so we can finish that pipeline. So after finishing the pipeline, we name it. Let's name it, I don't know, H, which stands for hierarchical. For that, we set the like, for example, k-means, and some of them are using the separation method. Some of them are using hierarchical representation methods. So they're of presenting results. They're all doing clustering in that repetitive way. For example, an algorithm to optimize model to separate parts of the data that we can explore. Its progress. We run a lot of pipelines here. For example, You can see all those pipelines, Bayesian networks, and everything that we run before. I might just go through some of these pipelines. Again, you have the pipeline for Bayesian network, and you can see the output files here, right? So uh, pipeline is generally finished in a couple of minutes. So we can create a pipeline for all those algorithms that we used so far. For example, we have the k-means, we have PCA. We also talked about that. We could have different outputs, output TXT outputs. We could have PDF plots, for example. And when opening those PDF plots, we could see some graphical representations of what we were analyzing. So I won't go in depth to all the steps of the pipelines right now because we don't have time for that. We also have some relevant issues. I just showed you how to uh, create a certain pipeline. And for example, I don't know, here we have also the RPAC, right? So I'm just going to download this RPAC into a specific folder, which is called RPAC. So let's see how to actually call the code in RStudio. So you just extract all the files in RStudio, first in that folder that you will actually open in RStudio. So let's go through directories and access that folder. It's right here, and we set this as a working directory, right? So we have three files. We have this call here. Let's open it, and we can copy the text from the code. Now, when I open the terminal here, you can see that my directory is not good. It's DCIS. So I'm just going to repeat the process and create, for example, a new terminal, which is now RPAC, right? So now it's good, and we can run the call. So you can see that the code is being executed, and that code that is executed is actually this script that we have in the output of the results. You can see the script here. You can see each part of the script that is used for different functions, read and prepare data. You have parse centering, main computation. This is something that produces the PCA results for us, and then we have draw PCA results as a PDF plot. So here's the PDF plot, and we can review everything in the same manner. So I'm not going to go through all the steps individually. There is also another important here that is used to analyze transcriptomic data. You have a demo pipeline, for example. Let me just go here on this button here. You have, for example, a cell line project, the same one we use to perform unsupervised and supervised machine learning. So you can review how to create a pipeline for an SRA file or those GTF and PESTA files Elia was explaining in last session. So you have the exact instructions as these yellow buttons here, how to create the pipeline. You have also explanations on each of these steps, what each step does. For example, you have these algorithms like cufflinks and cuff merge that deal with isoform construction and GTF processing, and you can finish the whole pipeline in the same way. So I'll just finish the pipeline without going in depth to the functions of each of these algorithms. And in the end, we create an expression table. We can run some other analysis here, and the expression table would have some output files. So we could have, for example, a txt file that is not filtered. In this case, we also have isoform files. We have stats table and many other files. So I'll just wait for this one to be downloaded, and you can review a txt file, tab delimited file of those gene expression levels. This could also be opened in Excel. So before we move forward, let me also review some other things that we were talking about. So we were talking about, for example, clustering. I will just open this PPT file. We were talking about different methods that are used on T-Bioinfo platform. So I'm just going to try to find PCA as well here. PCA PPT. Okay. So we were also talking about different types of data sets. And generally, we are analyzing data sets with a large number of genes, samples, big and heterogeneous data sets with complex relations between variables, between genes, samples. So generally, this is a lot of features, a lot of dimensions, and we are actually using machine learning to simplify this type of complexity. Why? Because large dimensionality, large complexity produces a lot of noise. There is a lot of irrelevant data for us, so we want to use some algorithms like PCA to actually reduce this curse of dimensionality for us. So we are creating principal components. We have that same pipeline on T-Bioinfo that is actually producing results for us. So in essence, we are creating a result like this that produces some separation between samples for us. Generally, each of these samples is separated based on all of those genes that were assessed in the PCA. So PCA is actually finding parts of the data that produce most variability and outputs them as principal components so that we could find subgroups or subtypes of breast cancer, for example, using this separation, all based on those principal components that are best for separating the data. So we also covered clustering, which is different from dimensionality in a way that it's actually not just finding parts of the data that are best used to separate data. It's not just producing principal components, it's also clustering data in separate groups or separate clusters. So clustering in some loose definition is actually grouping data based on their similarity, grouping parts of the data based on their similarity. So objects that are most similar will belong to same cluster, and they are different from objects that are in other clusters or they're not similar to that. So here, for example, we have an example of something that is similar to PCA in the sense that we're creating the plot based on principal components as well, but we are also circling out some clusters. We can see that this is definitely separated from this. Even though that, for example, we could say that this is a separate cluster, this, but you can see that those confidence ellipses just separated things here. So we are adding a statistical significance aspect to the analysis. For example, here we have a combination of PCA and clustering to improve the accuracy of the analysis. This was actually a k-means pipeline, if I remember well. So we also said that there are different types of clustering: hierarchical, partitional. We used some of them. We created pipelines, added the same call in RStudio, and actually run the k-means there. So we were able to produce hierarchical clustering as well, improve the visualization by customizing the plot and finding a variety of different subtypes of DCIS, if I remember well. in this case. So then we went to decision trees. Let me just see if I can find that one here. Okay, so we also have different aspects of analysis when viewing our tables. So we said that difference between unlabeled and labeled data is not that unlabeled data has no labels in general terms. So unlabeled data could have labels as well, could have names, but these labels are not used to categorize data to create classes. So your data could have names and still be unlabeled in a statistical way. So for example, here we have such a data set. It has IDs, that normal and SRRs are just part of the name. They're not classes. But if we were to label this data, it would have this uh, class row here. So you can see that it's categories. We have a distinct categorization between normal and basal and luminal B and AJR2 subtypes. What is very important is that just adding a normal to these names up here won't make it labeled, because still these identifiers will create unique IDs for all of the names. But if we have non-unique names here, they're non-unique, we have four normals which are absolutely the same, then classes or categorization is created and we have a label base. So labeling data is very important because, for example, in supervised machine learning, we need to have label data. Even in unsupervised, it's good to have some labeling in the data so that we can improve it or test it or do some other analysis. So we were speaking how to use label data, create pipelines on T-by-Info platform to create a decision tree models, which are actually predictive models with these cutoff values. So we can use this yes or no or cutoff value model to actually create predictions from a variety of genes, for example, to identify samples based. So we had an example that using these two genes, actually we can identify normal luminal A, luminal B, and AGI. So we won't go in depth there. We were talking about how decision trees work and everything. So we won't go there in detail in this moment. So we were also using other methods like LDA, QDA, support vector machines, which essentially did a similar thing, created a classification and predictive model for us. They were different in a sense, but they were also similar in this methodology aspect. So there are some differences in methodology, but in essence, they're also creating predictive models for us. So we use multiple predictive models to find out which one works best, which one should we use on those subtypes of breast states. Now, I will also repeat some things here. Elia showed you last time on some example projects that you can use, for example, to access use TCGA or Ensemble or other, which is uh, to provide just to data at this moment, but a lot of things like methods or literature. What are you looking? Just set all databases, but so you can go, for example, to GL with an omnibus. So where you can I don't know, you can type lung cancer here, for example, and let's see if we can find some dates. So generally, you will have a large number of data sets for pretty much uh, all things of interest that you would be looking for, because NCBI is very enriched as many other databases in large amounts of data. There are over 100,000 bio projects that are being performed with different types of data just here. So in many other platforms, there are different types of data and different types of bio projects. So generally, data is very important aspect here. So from my perspective, the most important aspect of conducting your project is actually having good data. So a lot of those things will work good. A lot of those machine learning methods will work perfectly if you have good data. But if you don't have good data, then you might struggle with using some of those machine learning algorithms. So for example, I don't know, let's just review some of these uh, data. So when you open this, you will have type of data here. So for example, we have type expression profiling by high throughput sequences. So let's go to that one, for example, just review. So we can review how was it performed, what are the platforms. We have Illumina here. You can also review many other things. So there are some things here that you could download. You have different formats, and we were talking about, for example, just picking it here and downloading a summary, right? So you can, for example, download a summary. You can also download uh, some supplementary materials here. So, for example, you have, uh, for example, these, and you can find, for example, microRNA counts here, or you could find FASTQ files, or here you have raw data are available in the SRA format. So we can download that. I don't know. Let's, for example, download this file. Going back here, you have soft formatted family files. So this is another important aspect. You can download this file as well. Let's just review those files. Let me just see if I can open it here. So generally you have a data set of those microRNAs. So Elia was showing you how, for example, to download a data set and convert it to transcriptomic gene expression table. Here you have, for example, different tumors of lung cancer, and you have some data on their microRNAs. So there are different types of data. It could be microRNA, it could be RNA-seq, it could be VCF file of variant mutations. Let me just get back here. Wait. So let's review this soft file, which is also important. What this file contains is actually some data that could be used to create a label. So, for example, you can see here sample characteristics, right? So you have genotype, right? So you have this AGO2 halo, you have normal lung, you have two genotypes, right? So you also have this another strain, right? So you have different, generally these parts where it says sample characteristics, you will have different labeling. Here we have CRAS-LS. We know that CRAS is a known oncogen, so it's marked by that gene. We also have here tissue. We have information that it's P53 null lung adenocarcinomas. So a lot of this information could be used, for example, to label the data. Let's see, for example, on some other transcriptomic example. Let's just type, I don't know, breast cancer. So Single cell RNA. Okay, so we have here, for example, single cell RNA sequencing. So let's try that one, for example. Let's see what we have. So we have a raw file here, right? So it's 20 megabytes, and we also have that soft file. Let's see how this file would look like from the perspective of creating classes. So you also have a lot of information, and here you have, for example, sample characteristics. Right? So you have some cell markers, CD2 low or CD44 high, and you also have the tissue, right? So each of these samples that is within the data set will have sample characteristics. And you can use those sample characteristics together with the method that Elia shown you. When you open it, here you will see, for example, where they're, for example, taken from some mouse model. They originate from some tissue, as you saw last time. And you have also cell specific, you have cell lines, you have molecular specific, and all those levels could be used to create classes. Or let's review another one. For example, we have another profiling here, but it's array. We will, for example, use this one. And let's see this file. You can see this one is much bigger. It's 3.6 gigabytes, the raw file, but the soft files are just a couple of information, which and this made it also here. So actually, you have, for example, sample character, the sample, and then when you another one which is also MCF7 but has differences, and you have 7 in this. Some slight differences. So in this case, would be a way to mark uh, different antibodies like FOXA1 here, and then we have, for example, we have this one, right? Different antibodies are reacting to different samples, so we can use those antibodies to label the sample, right? So there are different types of labeling, and as I said, some of them are tissue specific, some of them are antibody specific, some of them are biomarkers, some of them are just genes, some of them are just some representation from the clinical studies of how tumor looks, what is its size, what is its shape, what is the tissue that it's originating from. In the case of DCIS, ductal carcinoma, breast cancer carcinoma, we had the information that it originates from ducts of the mammary glands. And if it originates there, there's a high possibility that it's going to be a precursor for that invasive breast cancer. So location is sometimes very important from the biomedical perspective. We also have other platforms like Byron. I won't go with through the transcriptomic download Elia showed you in the last session, so I think that should be in your memory from that perspective. So I just showed you some reviewed some materials on how you can use metadata, for example, to combine it with the gene expression tables and all that NGS data that you can also download if it's publicly available from these datasets. Another important notion is that this data is generally, there is a lot of publicly available data, but there are certain rules on how to use it. For example, it could be used for research purposes, and there are guidelines on NCBI, so I recommend you to read them, not just on NCBI and also on other platforms, on the terms of usage of these publicly available data. So there are specific areas like academic and research areas that this data is available. So we have, for example, Firebrowse. So you can also explore this website. Oh, I'm sorry, it's on the maintenance. So I'll just go to ensemble, right? So different you also have different parts of the ensemble database that you can use to access the data. So you also have some options here on how to browse through species. What do you want to find? For example, you have here name or excision number that you would type here, and you would also get relevant information. You can also find single nucleotide polymorphisms here. So you can, for example, review not just transcriptomic data, not microRNA, not numbers, but you can review those things combined with, for example, variations in the DNA sequence. So, for example, here you have single nucleotide polymorphisms that are presented here. You have variant ID, you have chromosome number, location, everything, and what base is being substituted with what base. So you have T-C, which means that thymine is substituted with cytosine here, for example, and you have a lot of information here, some clinical significance, a lot of information could be explored here as well. So there are different platforms, and since I've been talking a lot so far, Aliyah can also, together with Mohit, explain you a lot of other aspects. So I'll just stop here and uh, let Mohit take from this moment and show you some aspects from his perspective. So I'll just unshare my screen. Okay. Mohit, you there?
Yes, yes, Darko.
Okay.
Yeah, sorry. Tell me.
Okay, so I ran through some things that we were doing so far, some recapitulation. So is there something that you would like to add here to the students?
No, I think that was very nice, and also the data sets you looked into, that was very thoughtful. And I think many of the participants were facing some issues with the data sets. So Vamshi had a few questions. So maybe if the participants have any questions regarding the same,
Okay,
we will.
so yeah, I see some people have issues with where to start the form, and so maybe we could post a link from the form in the chat on how to access the form, actually.
Sure. Let me do that.
Okay, great. So you have the link. Okay. So I also see that some of you have confidential data. So you don't need to share that at this point. You can contact Elia for that, and you can speak with him about that types of data. Another thing that I would like to mention to you is uh, to review that form that Elia has sent you. So you have different parts of the project. I'll just share my screen. Wait a second. Okay, so let's just, I don't know, we can go to any of these publications on NCBI. We can, for example, go to this one, and you will see that it has some summary, right? So this summary here is a good way of showing you how your abstract should look like. Not exactly like this, but it has some basic information about form, right? So you will have your own text, but what summary is actually containing is here you see, for example, to investigate downstream targets of that, that, and that, right? So that is the goal. Elia was speaking about goals last time. So that is the first and most important thing. What is your goal? What do you want to analyze? What are the results, potential results that you could expect? And what are these results related? So that is one of the things that should be contained in that project abstract that you're sending. Project abstract should be very concrete. You don't need to go too much in depth there. You just need to show the importance of something, importance of the data that you analyze. It could contain maybe some relevant information on why is this gene important, or why is this disease important, why is this data important that you analyze. So you also have some things about the overall design of the study. What is this? Where is this data originating from? What it contains. For example, it contains gene expressions, or it contains uh, mutation variants, or it contains single nucleotide polymorphisms, or it contains images, right? So define data, define goals. And third thing you should define are the methods that you're going to be using. Those methods, we are just giving you some ideas. You don't need to use just these methods. You could combine them with something else that is prior to your knowledge. But here we were giving you various ideas on some methods that you could include. For example, hierarchical clustering, or k-means in unsupervised analysis, or decision trees and random forests in supervised analysis. You could also use LDA, QDA, or support vector machines. Now, deep learning is a very popular method of analyzing large data sets today. It's also used in other aspects and other industries as well, for image recognition and text-to-speech recognition, many other industries. I know some of you have been asking me, can we use deep learning? In this moment, not, because it takes a lot of time to actually go through deep learning. So these machine learning algorithms have a lot of power to process pretty much any genomic, transcriptomic, or pretty much most of the data sets that you can find. Deep learning is used also in those cases, and it has some additional features like being semi-autonomous, sometimes even mostly autonomous, and it's very accurate results in image recognition. But as I said, if we were to use it in those areas, it would take much, much more time, like 6 or 7 months, to actually grasp some useful hands-on projects regarding the deep learning. So we won't be using deep learning in this program, but we covered it in some theoretical way just to see it as another perspective on machine learning. So you should define your project abstract. It should contain relevant information about the data, about the goals, about what you're analyzing, and about the methods that you'll be using. So, for example, you'll be using transcriptomic data, and you'll be using chemomatic and PCA TCR clean algorithms, KSAT algorithms, and that whole pipeline, or you can just name the pipeline. You'll also be using PCA. I'm just giving you an example. PCA and k-means, and you'll be combining it, for example, with hierarchical clustering if you want to do exploratory project. If you want to do predictive project or to identify genes that could be relevant to some disease, you could use a naive Bayes algorithm, you could use uh, discriminant analysis algorithms like these that we have here, or you could use decision trees and random forest. So whether you're performing an unsupervised analysis project or supervised, you should define methodology based on what we studied so far. So I also had a question from some of you whether you could use SPSS. Of course, you can use any program. You can use Excel, you can use RStudio, you can use the Bioinfo platform. The best thing to do, in my opinion, is to combine them. You can use SPSS as well. They're all powerful programs, which, for example, SPSS contains in its integrated interface most of these algorithms that we also use here, but in some quite a bit different form. So, in my opinion, you can use combine all of them, and that is the best way to actually perform the analysis. So there are some things that you should define maybe before our next session. Mohit has sent you the link to that form, and you can define these things that we mentioned. So you can define abstract, it should be just a couple of sentences. You don't need to write a whole essay on what you're going to do. Just a couple of sentences. If that is interesting to you, you don't have to do that, right? That is an option from the program, and we'll be here to guide you uh, actively through these couple of weeks, and then we'll also be there for another month regarding all the issues with your data set. So there is some limited amount of time that will be there for you to actually actively guide you through this process. So that's why we're kind of insisting on submitting your project abstracts and data sets so that we could work more on them during the duration of the program. So, okay, so Vamshi would like one-on-one session. Okay, anyone can have one-on-one session with us. So you can have it. I see some of you already have it with Aliyah. You can also have it with me. So I'm sure with other members of the Pine Biotech team. So please, you can send us messages. I'm going to leave you my email here. I've been leaving this a couple of times. But another thing that I would like just to note, you can contact us regarding any of the subjects that we were covering, but it would be good to stick to what we're covering in the program. We can go to some diversity, right? So we don't need to be exactly in these methods. You can ask absolutely anything about any other methods, but program is about biomedical data and using these machine learning algorithms and different aspects of the platform to process that data. So it would be really most optimal way of submitting this data and this abstract if they were related to what we are covering. For example, I don't know, you can post some completely other type of data that uses completely other methods, and we can also discuss that. But it would be professional, personally, both from my side and from the team, I believe as well, to actually uh, stick to these methods that we were using. We need to kind of we covered these with the hands-on examples, and I believe that you gain some knowledge related to these algorithms. So in my opinion, it would be best to actually, you don't have to use just transcriptomic data. You can use DCF files, you can use, as I said, those variant code files, other mutation files, and SMP files, polymorphisms. You can use different ways of classifying data. You can use non-normal, normal data. All types of data are there. But it should be relevant to biomedical industry, since that is something the program is covering. So that is just another notion that I mentioned. Please feel free to ask any questions, unmute the microphone, or you can post it in the chat. Okay. Any questions?
No, no question from me. Eli?
No questions. Okay. Others? You can feel free to share anything. If you don't understand anything, I've seen that some of you have issues with data sets, with the tables, with accessing the data. Generally, there are different file formats. Since you're not asking questions, I'm just going to go through this. So, for example, you have FASTQ file, right? So let's just go here. I'm just going to select any image here. You have FASTQ files that have sequences, some information, identifiers, a lot of information. For example, it's used in transcriptomic data. And we were analyzing FASTQ files in that first cell line pipeline to actually create an expression table. So in the end, you have an expression table that looks like, I don't know, like this. So you also have other formats. For example, you have on the NCBI here when you select nucleotide, I don't know, let's just type cancer here. You have different formats. So for example, you have SRA files that we used before, but you also have different formats. For example, let's select this one and see other formats that we have. So you also have that GFF or GTF file that Elia was explaining. You also have FASTA file. We said that both of these files need to be downloaded and uploaded to platform to actually produce that gene expression table. Let's just create this FASTA file and see how it looks like. So we can open it in Notepad, for example, and you can see that this is the FASTA file, right? So it says that it's messenger RNA, it's Homo sapiens family with sequence similarity 9-member B. So we have some mark here, some identifier, then we have the sequence. So this is the sequence that needs to be added to the platform to map it on a genome or transcriptome or other parts of the sequence files. Also here you have the sequence. Now this sequence that you're seeing here is, for example, for some identifier for some gene, but it's not universal, right? So for the same gene, this sequence could have variations. So some of these keys could be ACE in the same gene, in the same species, right? So this is just a model sequence. It doesn't have to be 100% same always. So keep this in mind that this sequence is used for mapping and it's varying. And you can see those variations when downloading, for example, SMP files. Let's review this file, GFF, right? So we were going through it last time as well. And if you open a GFF file, in Notepad, you can't really see a lot of things, but you can open it in Excel. And let's see how it looks in Excel. So in Excel, you have a lot of information here as well. You have the region, gene, CDS, exon. So you have information on where is this data derived from, whether it's intron or exon, whether it's a sequence, whether it's a gene or region. So this is also very relevant information. We have pluses here, which is telling you about the orientation of the DNA double helix. We know that each double helix in the DNA has a plus and minus strand, right? So it's telling us which strand is information derived from. The most important thing of GTF files is where the sequence is starting. So you have that information here, numbers where each sequence starting on what gene, what chromosome, and everything else. So you also have type of the data ref-seq here. So GFF or GTF files are containing information relevant to what is the start of the sequence in a genome. It's marked as numbers. And also you can see various numbers here. You also have information here. You have plus and minus strand and how it's annotated. What is the form of this sequence where is extracted from. So it's not saying if it's extracted from an exon or from a gene. Right? So it's very different because a gene could also have those non-coding regions which are between the coding regions, non-coding in a sense that they're not transcribed to proteins, they could be transcribed to messenger RNA and then cut. And then we also have introns and exons, so it's completely different type of information. A lot of this information is used, for example, to create a gene expression table on the platform. So you need a variety of formats to actually produce that gene expression table. But you also have other files here. For example, let's review the SNPs here, and I believe that when you open the SNPs, you need to specify a gene here. So, for example, let's find some gene. Let's go to insulin, right? So you have a lot of information on insulin here, right? So it doesn't actually have SNPs, but it has variants, right? So let's review them. So you have this clinical assertion, whether it's pathogenic or not, what is the clinical significance here. So you also have information that could be used in classifying the data. So you have likely benign, non-benign, pathogenic, and that kind of information could be a useful input for the analysis. So you have the study ID here as well. You have the region ID. So you have type. You have a lot of information here. You can access any one of them. For example, let's access the region here. Let's see how it looks like. So it will just load in a couple of seconds here, and you will have those regions. Now it's quite complex here. You have the chromosome where it's located. You have regions where this polymorphism